Test clusters. An organism that shows a dominant characteristics can have either of two possible genotypes. It could be homozygous for the dominant allele, or it could be heterozygous. For example, a gray chinchilla could have the genotype B, G, or small and B. We can find out the genotype of an individual of the dominant phenotype for a particular gene by crossing it with one known to have the homozygous recessive genotype for the same gene. This is called a test cross. For example, if we know that the allele for tallness is dominant to the allele for dwarfness in a certain species of pea, then the genotypes of any tall plant can be determined by crossing it with a dwarf plant. If any of the offspring are dull, then this will mean that the tall plant has an allele for dwarfness. It must have been heterozygous. If none of the offspring are dwarf, this more certainly means that the tall plant was homozygous for the tallness allele. However, unless there are large numbers of offspring, this could also happen if the tall parent is heterozygous, but just by chance none of its gametes carrying the recessive allele were successful in fertilization. So, pure breeding. Some populations of animals or plants always have offspring just like themselves. For example, a rabbit breeder might have a strand of rabbits which all have brown coats. If he or she interbreeds them with one another, all the offspring always have brown coats as well. The breeder has a pure breeding group of brown rabbits. Pure breeding organisms are always homozygous for the pure breeding characteristics. Heterozygous organisms are not pure breeding. Sex determinations. Right at the start of this chapter, we look at the complete sets of chromosomes in a human cell. One of the pairs of chromosomes in figures is responsible for determining what sex a person will be. They are called the sex chromosomes. A woman's sex chromosomes are both alike and are called X chromosomes. She has a genotype XX. A man, though, only has one X chromosome. The other smaller one is a Y chromosome. He has a genotype XY. Even without sex, inheritance endures the same way as for any other characteristic, but using a letter symbol to describe whole chromosomes rather than individual alleles. Okay. Offspring genotype phenotype. So, each time a child is born, there is a 1-to-1 chance of it being either sex. Sex linkage. The X and Y chromosomes do not only determine sex, they have other genes on them as well. We are seeing that for most chromosomes, we have 2 copies of each one. A homologous pair. They contain the same genes in the same position. This means that we have 2 copies of each gene. But this isn't true for the sex chromosomes. The Y chromosome is tiny and only has a few genes. The X chromosome is much larger and has many more genes. This means that for most of the genes on the X chromosome, man only has 1 copy. There's no second copy on the Y chromosome. So for most of the genes on the X chromosome, a man only has 1 copy. There's no second copy on the Y chromosome. And then genes in this Y chromosomes are not found in X chromosome. Sex-linked genes are found in this part of the X chromosome. There are also a few genes on the Y chromosomes that are not found in X chromosomes. This means that a woman never has a copy of these genes and a man has only 1 copy. Genes that are found on the non-homologous parts of the X and Y chromosomes are called sex-linked genes. One of these sex-linked genes controls the production of the 3 different kinds of chromosome cells in the retina. A recessive allele for this gene, small letter B, results in only 2 types of chromosome cells being made. A person who is homozygous for this allele can cannot tell the difference between red and green. They are said to be red-green colorblind. This condition is much more common in men than in women. It shows the various genotypes and phenotypes for this sex-linked condition. You can see that there are 3 possible genotypes that a woman might have, but only 2 possible genotypes for men. When we write genotypes involving sex-linked genes, we need to show the chromosome as well as the line. So, the 5 possible genotypes and the phenotypes for red-green colorblindness are SB woman with normal vision, SB and then S small letter B, woman with normal vision who is a carrier, S small letter B, woman with red-green colorblindness, S big letter B and Y, man with normal vision, S small letter B, Y, man with red-green colorblindness. Inheritance of sex-linked characters. We can use a genetic diagram to show how sex-linked genes are inherited. For example, is what might happen if a woman who is caring for red-green colorblindness marries a man with normal vision. Normal man is SB1, and carrier woman is SB. So SB or Y, SB or SB, and then offspring genotypes and phenotypes. So you make it into genetic cross where SB, then SBY. So SB, big letter, small letter, SBY, and then SBY also. So you get normal female carrier, normal male, red-green colorblind male. This genetic diagram predicts that about half of the male children will be red-green colorblind. Half of the female children will have normal vision. Genes and protein synthesis. Chromosomes are made of DNA. In chapter 4, saw that a DNA molecule is made out of 2 long strings of molecules called nucleotides. These are 4 different nucleotides, each containing a different base, A, C, T, or G. This figure shows the structure of a very small part of a DNA molecule. You have seen this diagram before in chapter 4. And you should also remember that protein molecules are made up of long chains of smaller molecules, amino acids. There are 20 different amino acids, and the sequence of the amino acids in a protein molecule determines the final shape of the molecules. So, this shape affects how the protein works. So, for example, the sequence of amino acids in an aminase enzyme determines the shape of the active site into which a starch molecule can fit and form an enzyme substrate complex. If the sequence of amino acids is not absolutely correct, then the shape of the enzyme will not be correct. Its active site may not be a complementary shape to a starch molecule, and it will not be able to catalyze the reaction in which starch is broken down to maltose. A gene is the length of DNA that codes for making a particular protein, for example, amylase. The code is in the form of sequence of bases. The sequence of bases in the DNA molecules determines the sequence in which amino acids are linked together to produce a protein. The sequence of bases in DNA molecule determines the sequence in which amino acids are linked together to produce a protein. In this way, the DNA in the nucleus controls the functions of the cell. It does this by determining the shapes and therefore the functions of all the different proteins that the cell makes. These include not only enzymes but also other proteins such as the carrier proteins in the cell membrane and the receptor proteins onto which neurotransmitter molecules can bind and assemble. DNA is found in the nucleus. Protein synthesis happens in the ribosomes in the cytoplasms to carry information from the DNA to the ribosome messenger molecule called mRNA is used. When a protein is to be made, carefully controlled sequence of events take place. First, an mRNA molecule is made in nucleus, copying the base sequence for a line of DNA that makes up a gene. The DNA stays in the nucleus. The mRNA moves out of the nucleus and into the cytoplasm. The mRNA then attaches to a ribosome. If you have been eating a good diet, then the cytoplasm in your cells will contain plenty of all the 20 different amino acids. As a long-chain mRNA molecule passes through the ribosome links amino acids together in exactly the right order to make the desired protein, following the code continued on the mRNA molecule. So every cell in your body contains tens of thousands of genes. All of your cells contain exactly the same genes because they have all been produced by mitosis from the original zygote. They are genetically identical, but any one cell does not need to make all of the possible proteins that its gene code for. Each cell needs only a particular set of protein. For example, a cell in your skin might need to use a gene to make keratin of the protein that has makeup, and a gene to make the pigment that colors the keratin. But a neuron in your brain doesn't need to make either of those proteins. Instead, it makes proteins such as the receptors on its membrane that allows neurotransmitters from other neurons to bind to it and set up nerve impulses in it. When the information carried by a gene is used to synthesize a protein, we say that the gene is expressed. Each specialized cell expresses only the genes that it needs in order to carry out its specific function. Some cells, however, are not specialized. For example, when the zygote is first formed, it divides repeatedly to form a little ball of almost identical cells which make up an embryo. At this stage, you cannot see any differences between the cells. Each cell is able to divide to produce other cells which can then become specialized for different functions. These unspecialized cells are called stem cells. A uniform embryo contains nothing but stem cells. By the time we're adult, most of our cells have become specialized, but some stem cells will remain in our bodies. These are really important to us because they are able to divide by mitosis to form other specialized cells. For example, we have stem cells in our bone marrow that are always dividing to produce the specialized cells in blood, red blood cells, platelets, phagocytes, and lymphocytes. Stem cells in the brain can divide to produce new neurons. The range of stem cells that are present in an adult human has only recently been discovered by scientists, and we are still learning a lot about them. A better understanding of stem cells can lead to new treatments for medical problems that we currently cannot solve. For example, could stem cells be used to produce new heart muscle in someone with heart failure? Could they produce new nerve cells for someone with damaged spinal cord? Science is moving on quickly in this field of medicine research.